You don't call on me for a question today. I'm gonna try to do the best I can.
Okay. Or if I
That's
get,
why I let you know just in case.
if I get right in front of you and talk down at you. Can you hear me? Can you hear me right now?
I mean, yeah, it's just this my left ear that I don't have it.
Uh-huh.
That's my worst ears.
Okay. Yeah, can you hear me? And if you tell me yes, you'll probably have to unmute yourself. I think there's Jocelyn. Yeah, can you hear me?
Yes.
Okay, well. Good morning, everyone. So we've got some people out sick. Last week, it seemed to me there was some sort of enterovirus infection going around campus. It was some sort of GI thing going around. This time, it seems like it's some sort of upper respiratory something going around. So I'm not sure what the deal is, but it seems like folks are getting sick. So do your best, cover your cough, wash your hands, eat your green beans, you know, that sort of thing and all that. Yes, your mother was right. Try to stay healthy. Your lab books are in the office yet again. And I think I left my water bottle there too. So I'll try to negotiate this without drinking any water, which is going to be hard to do. All right, so we're on metabolism for today. So we're going to use our little makeshift blackboard here because folks are on Zoom. I want them to see what's being written. So we talked before about the role of ATP. We'll go now from the role of ATP into the actual pathways. And remember, for these pathways, you need to know what goes in and what comes out. You don't need to know the actual steps in the pathway. The book talks about the steps in the pathway. I teach classes where students have to learn every step in the pathway, all the chemical structures and so on and so forth. That's not one of you. As a nurse, you won't care essentially what the structure is of phosphoenolpyruvate. Seriously, it won't help you as a nurse. So because of that, I'm not going to require that of you. Having said that, you need to know what glycolysis is and what it produces and what it works on. So as we said before, this is ATP. So if we look at ATP, so under physiological pHs, these molecules here, it says that they're hydroxyl groups. Actually, at pH 7, these are going to be oxygens with a minus charge on them. So you have 3 oxygens here with a negative charge. Now, what do you know about negative charges when you place them close together? Do they like each other? No, they don't like each other. They want to get away from each other. So this bond here and this bond here, these are called phosphoanhydride bonds. Phosphoanhydride bonds. And phosphoanhydride bonds have a lot of energy. So if you add a water across this phosphoanhydride bond, it's really hard to draw with a mouse. You add a water across this bond here, you're going to get inorganic phosphate over here, and you're going to get ADP over here. Both ADP and inorganic phosphate are capable of resonance stabilization. Resonance stabilization will take the negative charge and distribute it over the whole molecule. That's very stable. Because of this, your two end products are very stable. That makes the reaction much more likely to go. Because of that, you release bucketfuls of energy when you hydrolyze that bond. And yet, in the cell, ATP is relatively stable. Because of its relative stability, ATP is a good storage molecule for energy. So if you want to drive a reaction that's not very favorable, you couple that reaction to the hydrolysis of ATP. Many proteins that drive reactions that are not very favorable have pockets in the protein for binding ATP. We call those pockets Rossmann folds. A Rossmann fold has a very peculiar structure to it. And if you're looking at protein sequence structure, you can usually see one pretty easily if you know what to look for. But that Rossmann fold means it binds ATP really easily. And the hydrolysis of ATP, that energy released, helps drive the reaction. So ATP hydrolysis will drive a lot of your anabolic reactions, and ATP synthesis creates energy in catabolic reactions. So if we look at redox reactions, so remember redox stands for oxidation reduction reactions. Loss of electrons is what? The phosphorylation, oxidation, fermentation, reduction. Which is it? Oxidation, oil rig. Oxidation involves loss. Loss of what? Electrons. Rig. Reduction involves gain. Gain of what? Electrons. So if we move to catabolic reactions, our energy will be captured in the form of ATP. High energy electrons will be captured by these two carriers, NAD and FAD, and they will create these reduced electron carriers, NADH and FADH2. That's the other way you're going to store energy. You're going to take high energy electrons from compounds, oxidize them, but you don't want free electrons floating around in a biological system. Very dangerous. You give them to electron carriers. So this summarizes some of the ways essentially that metabolism is going to work. Now, the first set of reactions is going to be glycolysis. Glycolysis. Glycolysis will produce products that will go to the Krebs cycle. Krebs cycle generate these electron carriers You need to oxidize those electron carriers, and you'll do that in the electron transport chain. And the electron transport chain will produce lots of ATP. Glycolysis produces just a little bit of ATP. Fermentation is going to give you all your ATP via glycolysis. So fermentation is not going to give you that many ATP. Fermentation does not necessarily mean you make ethanol. There are lots of fermentations that make organic acids. They make things other than ethanol. But fermentation means that you don't use electron transport. That's the biggest part of a fermentation. You make all your ATP by a process called substrate level phosphorylation. Substrate level phosphorylation means you take a carbon compound, you make a carbon phosphate bond. That bond is high energy. You then transfer the energy of that carbon phosphate bond to ADP to make ATP. That's substrate level phosphorylation. That's the way you make ATP in glycolysis. If you make all your ATP via substrate level phosphorylation, you carry out a fermentation. That's what a fermentation means. So aerobic respiration means your terminal electron acceptor in the electron transport chain is oxygen. You reduce oxygen to water. That's aerobic respiration. So you use these other pathways, you use them to generate lots of reduced NAD, lots of reduced FAD. You need to reoxidize those carriers if you're going to oxidize further carbon sources. To do that, you give those reduced carriers to an electron transport chain. Those high energy electrons go down the electron transport chain and they're given to oxygen to make water. You don't have to use oxygen. Oxygen, as electron acceptors go, is the best. But if oxygen is not available, you don't have to use them. So several of the soil bacteria that are in that soil right now, if that soil were to get inundated with water, once all the water gets used, once all the oxygen gets used up, these organisms don't have to use oxygen. They can switch to nitrate. They can reduce nitrate to nitrite and ultimately to nitrogen gas. Nitrogen gas is re-released into the atmosphere. Ecologically, that's called denitrification. You can also use sulfate and reduce sulfate to sulfite and elemental sulfur or to sulfide. And you can use other molecules too, but you don't have to use oxygen. We have to use oxygen. We can't adapt to nitrate or sulfate or something like that. Many bacteria can. Many bacteria can. And then fermentation is going to use the carbon skeleton and it's going to dump reducing equivalents right back on that carbon skeleton. That's why fermentation will always release reduced carbon compounds. Ethanol is a reduced carbon compound. Lactate is a reduced carbon compound. Butyrate is a reduced carbon compound. Butanol is a reduced carbon compound, so on and so forth. All right, so let's now talk about glycolysis. I have to keep remembering to go back to, all right, so let's use our camera here. Light on, lights on, all right. Can you please shut these lights off? The glare on that screen is ridiculous. Better? No, just that one, I think. I don't want you guys falling asleep. You haven't had your coffee, you know, it's pretty bad. All right, so with glycolysis comes from two words in Greek. And glycos in modern Greek is used for dessert. Well, it's in a lot of desserts that makes them taste sweet. Glucose, right. Glycos is essentially how we get our word glucose. The other word, lysos or lucis, means to destroy. So this is the pathway that destroys glucose. So glucose is a 6-carbon compound. I'm going to draw the structure of glucose. You don't need to know the structure of glucose. I'm just going to draw the structure because it'll make it easier to show you how this works if I draw the structure of glucose. All right, so that's glucose. Glucose undergoes sequential series of modifications. Every step is enzyme catalyzed. The first reactions consume ATP. You consume 2 ATP. You break glucose into 2 3-carbon compounds. So then the pathway diverges. And eventually you're going to produce 2 ATP over here, 2 ATP over here. You're also going to make a reduced NAD. And you're going to eventually get out of this pathway a 3-carbon compound called pyruvate. So you get 2 of those. These are 3-carbon compounds. So you started out with 6 carbons. You go down here, you have 2 3-carbon compounds. How many carbons did you lose? Well, Morgan's from Arizona. She likes the sun, but I don't think she likes it that much. How many carbons did you lose? Okay, see, some of you are doing that. You didn't lose any, did you? So you don't lose carbons yet. So pyruvate is the end product of glycolysis. What goes in? Glucose. Now, do you have to use glucose? Can you use fructose? Can you use galactose? Can you use mannose? Yeah, you can. You can use other 6-carbon sugars. So a 6-carbon sugar, glucose is the most popular, shall we say. What comes out? 2 plus 2 is 4 minus 2. So how many ATP? Gosh, focus you. 4 minus 2 gives you how many net ATP? 2. So 2 ATP. How many reduced NADs you get? 2. Glucose went in, and then how many of these pyruvates do you get? 2. And the book calls them pyruvic acid. That's a non-ionized version. At physiological pH, they're going to be ionized. That's why I call them pyruvate. Pyruvate, because the structure, again, you don't need to know this, but the structure of pyruvate looks like this. Carboxylic acids, what do you know about the pKa of carboxylic acids? pKa's of carboxylic acids are about what? Who thinks 1? Who thinks 2? Who thinks 3? Who thinks 4? Who thinks 5? Who thinks 6? Who thinks 7? How many have no idea what a pKa is? pKa tells you the tendency of this to ionize. The lower it is, the more likely it is to ionize. The higher it is, the less likely it is to ionize. This is a carboxylic acid. Its pKa is somewhere between 3 and 4. What's the pH inside a cell? 7-ish. This is below 7, so it's likely to be charged. It's likely to be ionized. That makes it pyruvate instead of pyruvic acid. You put the hydrogen on there, it now becomes pyruvic acid. All right, so that's glycolysis. That gives you pyruvate and these molecules here. Okay? Gracie, what goes into glycolysis? Glucose. Lexi, what else can go into glycolysis? Okay, ATP, sugars, wise. What else can go into glycolysis? Fructose. What else? You'd have to degrade it for lactose is a disaccharide. You'd have to degrade it to monosaccharides. Lactose gets degraded to which monosaccharides? You're mumbling. I can't hear you. Yeah, galactose and glucose. Can galactose enter glycolysis? Not directly. It has to be converted to glucose first. But it can. So it can, but indirectly. Okay, Morgan, what comes out of glycolysis? Name one thing that comes out of glycolysis. 2 pyruvates, 2 pyruvic acids. Hannah, what else comes out of glycolysis? 2 ATP. NaOH is a base. NADH. NaOH is sodium hydroxide. Very poisonous. No, you don't make sodium hydroxide. It's, yeah, I know my handwriting is terrible. NAD, that's a D. D. Looks like an O. Yeah, D. And NADH is nicotinamide adenine dinucleotide. And Devon, nicotinamide is derived from dietary. Nicotinamide sounds like niacin. Niacin. Say it. Niacin. This is the reason why you need niacin in your diet, to be able to make NAD. Okay? Niacin. All right. And Kara, you're next. All right. What then happens to these pyruvates? Now, if you ferment, you're going to take these NADHs, you're going to give those reducing equivalents to either pyruvate or a derivative of it. That's essentially how you end up with ethanol, which is a 2-carbon compound. Because ethanol, you take the carboxylic acid off this group, you make carbon dioxide. That gives you acetate. You reduce acetate, you get ethanol. The other thing you could do is just reduce this here. That gives you this molecule here. And this, my friends, is lactate. Ever heard of Ringer's lactate? If you haven't, you will, because you'll be hanging Ringer's lactate bags for patients. That's what's in the Ringer's lactate. That's lactate. And that's what gets made by muscles, aerobically active muscles, when they're working really hard. And they undergo lactic acid fermentation. That's what you make with lactic acid fermentation. Okay, that's glycolysis. Now, if we go back to our slides. Pyruvate enters the Krebs cycle. The Krebs cycle is going to completely oxidize a derivative of pyruvate. So pyruvate first undergoes a reaction with a large enzyme complex called the pyruvate dehydrogenase complex. And I think it behooves me to show you this reaction on paper again. So there is a carrier inside cells. It's in bacteria, it's in our cells. It carries carbon chains. It's called coenzyme A. Coenzyme A requires the vitamin pantothenic acid. And sometimes if you read the back of milk cartons, like you might do with your breakfast cereal if you're bored, you read the back of the cereal box or the milk carton. It says some milk types are fortified with pantothene or pantothenic acid. This is a chemical derivative of pantothenic acid. These are used in our bodies to make coenzyme A. That's why you need pantothene in your diet. So coenzyme A is used in this next reaction. You take pyruvate, coenzyme A is fed into this, and so is NAD. From this, you'll get carbon dioxide, and you'll get this part of pyruvate. This is acetate. And this will be linked to coenzyme A. Coenzyme A is a sulfur at the end of it, and that's sulfur. So this is called and this is written like this, and this molecule is called acetyl coenzyme A. Acetyl coenzyme A. The enzyme that catalyzes this reaction is, stop calling me. The enzyme that catalyzes this reaction is called, it's a big, huge complex in our mitochondria. Bacteria don't have mitochondria, so it's in the cytoplasm. It's called the pyruvate dehydrogenase complex, which that's kind of a mouthful. So let's abbreviate that. PDH complex. I like that better. Don't you? This is before the Krebs cycle because acetyl coenzyme A is the substrate for the Krebs cycle. So you have to make acetyl coenzyme A from pyruvate before it enters the Krebs cycle. Now, in our cells, pyruvate enters the mitochondria. The Krebs cycle occurs in the mitochondria. Do bacteria have mitochondria? Survey says no. So where does this occur? In the cytosol, in the cytoplasm. So you generated essentially from this another reduced NADH. So Kara, your turn. What goes in to the PDH complex catalyzed reaction? What goes in? Pyruvate. What comes out? CO2 comes out. Catherine, what else comes out? No, not ATP. NADH. And what else comes out? There you go, acetyl coenzyme A. Very good. Very good. And Jetty, where'd the coenzyme A come from? It's made from pantothenic acid, and then it's in the cell. So it came from the cell. That's a little bit of a trick question. That's a little bit of a dirty trick. But you're a good sport, so I can pull dirty tricks on you, right, Jetty? Sure. Okay. Okay. Acetyl coenzyme A, which we'll write like that, now enters the Krebs cycle. The first product of the Krebs cycle is citric acid. That's why this cycle also gets called the citric acid cycle. So acetyl coenzyme A will combine with a compound called oxaloacetate. This condensation will make citric acid. You will release the coenzyme A. The coenzyme A is recycled. And it goes back and it loads up with another acetate, comes back to the Krebs cycle. The enzyme that catalyzes this reaction is called citrate synthase. Absolutely fascinating enzyme from a biochemical standpoint, but that's all I'll say about it. Now, citric acid gets oxidized sequentially, and the sequential oxidation regenerates the oxaloacetate. And in doing so, how many carbons are in acetate? 2. Pyruvate, focus. Pyruvate had 3 carbons. Acetate now has 2. So you're going to give off 2 carbon dioxides. And you're going to generate a bucket full of NADHs. You're going to generate 3 of these, 3 reduced NADs. You're also going to generate a reduced flavine, and you'll generate a GTP via substrate level phosphorylation. Now, GTP, when you see GTP in your head, think ATP. Because GTP and ATP are interchangeable. So, for every turn of the Krebs cycle, what goes in? Acetyl coenzyme A. What comes out? 2 carbon dioxides, 3 NADHs, 1 reduced flavine, 1 FADH2, and 1 GTP for every turn of the cycle. Because each glucose gives you 2 pyruvates, you're going to do 2 of these. So per glucose in the Krebs cycle, you'll generate 6 of these, 4 CO2s, 2 NADHs, and 2 GTPs per glucose. Melina, you don't look too sure about that.
Can you repeat why there'd be two?
Because glucose produced 2 pyruvates. How many carbons does glucose have? 1, 2, 3, 4, 5, 6. Remember, glucose is C6H12O6. It's carbohydrate. There's the hydrate, there's the carbo. So C6, stop. Scam calls are enough to drive you to drink Mountain Dew. There's 6 carbons here, so you generate 3 carbon compounds. That's why when you get rid of a CO2 here, remember that there are 2 of these per glucose. So because there are 2 of these per glucose, you're going to have 2 of these, 2 of these, 2 of these, 2 of these, right? And this has 2 carbons because you got rid of 2 right here. So that's why you're going to have to run this reaction. You're going to have to run it twice per glucose. So that's why you'll get this times 2 per glucose. Okay, that better? Okay, since you understand so well, why don't you tell me what goes in to the Krebs cycle? Acetocoenzyme A, very good. Paige, name one thing that comes out of the Krebs cycle. 2 CO2s, very good. Kenna, what else comes out of the Krebs cycle? GTP, very good, very good. Gracie, back to you. What else comes out of the Krebs cycle? FADH2. Lexi, what else comes out of the Krebs cycle? NADHs. Very good, very good. Now, the cell has a problem at this point. And if you're thinking about it instead of just trying to catch up, this is actually a pretty sizable problem. So you started with this carrier, you started with this carrier. Now, catabolism generates this, and this. This is what you use to oxidize carbon compounds. If all your carriers are reduced, how can you oxidize more carbon? You can't. You have to re-reduce these. And that's what the electron transport does. The electron transport chain is a sink for these high energy electrons. So it's going to give these to the electron transport chain. In mitochondria, the electron transport chain is embedded in the inner mitochondrial membrane. In bacteria, this is in the cell membrane. The cell membrane. So the cell membrane is very busy in bacteria. It's a very busy structure. Very good. So this shows you the cyclical nature of the Krebs cycle. It allows you to count the carbons. You see the acetate combining with the oxaloacetate to make the 6-carbon citrate. You make 2 CO2s and lots of other reductant. It shows you there. And again, that gives you the math per glucose. Then we get to the electron transport chain. Okay, back to our makeshift blackboard here. For an electron transport chain, you need three things. You need an intact membrane. And that membranes are impermeable to charged molecules. So it must be impermeable to hydrogen ions, which intact membranes are. Next thing, you're going to need electron carriers embedded in the membrane. And those carriers have to have a specific set of properties. The carriers must be arranged in two ways. They have to alternate between those that carry hydrogen ions and electrons and those that carry just electrons. So it has to alternate between carriers that carry both hydrogen ions and electrons and those that carry just electrons. Secondly, they must be arranged so that the thermodynamic transfer of electrons is spontaneous. So they must, there's a, I don't know if you guys study this in GOB. Did you study this concept of delta E? That's a tendency for something to take up electrons or give up electrons. Okay, I'm seeing some nods. So if something has a delta E and that delta E is very positive, that means it really likes electrons. The more positive, the more electron hungry it is. The more negative, the more electron hating it is. So it must be arranged in such a manner that the delta E goes from negative to positive. That makes the flow of electrons down the electron transport chain thermodynamically spontaneous. So let's rehearse. You have to have an intact membrane. Embedded in that intact membrane, you need electron carriers. And then those electron carriers have to be arranged, one, so that they alternate between those that carry both hydrogen ions and electrons, and others that carry just electrons. And they must be arranged from most negative electronegativity to positive electronegativity. Let's talk a little about carriers. So if we talk about electron carriers, so those carriers that will carry both hydrogen ions and electrons, the most prominent component of these are ubiquinones. You might know these by a different name. Coenzyme Q10. It used to be all the rage, at least back in the 1990s. Athletes, particularly endurance athletes, would take coenzyme Q10 tablets before races in order to do better. I don't know if any of you who are athletes have ever heard of it or even tried taking it or what have you. No? No, well, I think people stopped taking it because they saw it really didn't do any good. But anyway, these are components of electron transport chains. These are very lipid soluble, so they diffuse through membranes very rapidly. These are also small molecules. That means they can move between larger protein carriers very easily. The second ones will usually be flavines. Flavines, and so they will be linked to proteins. Those are called flavoproteins. And Janelle, we remember flavines are derived from which dietary vitamin? It has flaving in the name. Add the sugar ribose and it becomes riboflavin. Riboflavin. It's a type of B vitamin. So niacin's a B vitamin, riboflavin's a B vitamin, thiamine's a B vitamin. That's part of the B complex. What kind of foods will be rich in B vitamins? Anything with yeast in it. Yeast are a rich source, so breads. Beer, although the fermentation tends to destroy a lot of the vitamins. Also, the distillation destroys vitamins. So beer, if you said beer, I'd say, no.
But when you're talking about niacin, what kind of foods have niacin in them?
Anything that is leafy green will be rich in niacin. Anything rich in yeast will be rich in niacin. So any of the yeast pastes, if you can tolerate them, will be very rich in niacin. So will all of your cruciferous vegetables will be rich in niacin. Spinach be rich in niacin. Other types of kale, things that people hate, things your mother told you you used to went, no, I don't want to eat that. Unless you're my middle daughter who ate all her vegetables and used to snack by plucking on salad. She still loves salad. So I didn't have to get her to eat salad. She would eat salad on her own. I guess you could say dietarily, she was a strange kid, I suppose. But those sorts of things are all usually quite rich in niacin. These are things that carry both hydrogen ions and electrons. Let's take a look at things that now carry just electrons. Things that contains cytochromes. Cytochromes have iron. Iron only binds electrons. So various types of cytochrome proteins. Also a group of proteins called iron sulfur proteins. Only carry electrons, these. So by arranging these in the membrane, there's our ubiquinone. By arranging these such in these large complexes, you can get them so that you have particular ones that carry just electrons, those that carry electrons and protons, those that carry just electrons, those that carry electrons and protons, those that carry just electrons. You then get your coenzyme Q10s here. This will shuttle things, say, to here. This will carry, these carry both electrons and hydrogen ions. These will carry just electrons, but they may have other ones in here, carry electrons and da-da-da. These then go to something else, and then ultimately, you're going to give these electrons to oxygen, and you're going to make water, like that. Now, something happens on the way to the bakery. As you pass these through the electron transport chain, this carries both electrons and hydrogen ions. This one doesn't. What happens to those hydrogen ions? They get sloughed to the outside. They get pumped, essentially, across the membrane. So as you transfer electrons down this chain, you're also pumping hydrogen ions to the other side of this membrane. You see why this membrane needs to be impermeable to hydrogen ions? A lot of you are looking at me and say, well, big deal. Big deal, gang, deal. Because now you're generating a disparity in concentration. You're generating a disparity in charge and pH. That can be used to do work. You're generating a big battery here. And that's precisely what the electron transport chain does. It creates this disparity in pH, charge, and concentration. This collectively is known as a proton motive force. A proton motive force. The proton motive force will be used to do work. Let me pause right there and see if there are any questions.
So proton motive force, is that hydrogen ions being pumped through that?
Yep. The hydrogen ions are being pumped across the membrane. They create a lot more hydrogen ions on this side of the membrane than on this side of the membrane. Because remember, the membrane, an intact biological membrane, is impermeable to charged entities. So these have to stay out here unless there's a door in the membrane to let them in. But if you put a door here, you can charge a toll for them to come in. And that's how the cell is going to make so much energy out of this. And that energy, potential energy created by the disparity in pH, charge, and concentration across the membrane is called a proton motive force. Is Paige the only one brave enough to ask questions, or? I realize there's a lot to throw on you, especially on a Monday morning. But understanding this is really important to how cells do business. All right, let's take our 5-minute break. Blow on your pens, make sure they cool off, and rest your heads, and then we'll come back and pick this up. All right, those of you on Zoom, if you have any questions, go ahead and use the chat function to submit your questions. Either that, or you can just unmute yourself. I'm next to the computer. You can unmute yourself and ask me right now.